Hi, I'm Luke Piggott. I'm Associate Principal Scientist in Translational Pharmacology and Translational Medicine Unit. So small cell lung cancer is a type of cancer that carries a high level of genomic instability and is frequently treated with DNA damaging agents, carboplatin and etoposide. Although the patients in the second line do respond to these drugs, responses are often short-lived, and there's a medical need to try and improve responses to these standard of care therapies. And what we wanted to do was to see if we could improve the efficacy of this drug combination and also see if it could be something that could be tolerable as well. Debio is a highly selective and potent WEE1 inhibitor, and what that means is that it doesn't have much activity on other kinases. What it does is it inhibits WEE1, which is an important kinase regulating the cell cycle, at both the G2, M and S phase checkpoints, which are usually there to repair damaged DNA in tumor cells, and by inhibiting WEE1 at those checkpoints, we can drive those cancer cells into mitotic catastrophe and ultimately cell death. First of all, it was interesting to see that we improved the responses in combination with just carboplatin or etoposide alone as a combination partner with Debio 0123. But then more importantly, when we combined them as a triplet, as a combination, we saw significant improvements in the tumor response over and above the standard of care treatment, which is just carboplatin and etoposide treatment alone. We also found that each of these combinations under the conditions that these studies were performed was well tolerated, which is very key for being able to translate these results into the clinic. The implications are that this sets the scene for the potential of WEE1 inhibitors, especially Debio 0123, which is clearly well tolerated from these studies preclinically, to put them into clinical cohort of patients.